This is Group THJ, and this is our presentation about how we analyzed our gene of interest and how we tried to determine its function.
Our gene was found to have 2 exons and 1 intron, which was confirmed by various softwares to assure there was not a mistake. Additionally, our ESTs were consistent with our findings. As for our exons, the first one was found to be on base pairs 515 to 1036, making it 521 base pairs long. The second exon was found to be on base pairs 2373 to 3305, which makes it 932 base pairs long.
We analyzed the phylogeny of our gene of interest and also of some related species of plants. Our phylogenetic tree has a high degree of confidence because of the high values between 98 and 100. The closest related gene is from Oryza sativa, and they were found to be 62% similar, which is not super similar, but it does indicate that they are related in some way. And all of these proteins were uncharacterized, including our gene of interest. However, we did find one transmembrane protein that was from Arabidopsis thaliana, which is shown on this phylogenetic tree, and we found it to be somewhat related to our protein of interest.
Overall, our gene was found to be uncharacterized, meaning that not much research had been done on it. As you can see on the slide, the closest relative to our gene is a transmembrane protein in Arabidopsis. However, it was only found to be 49% similar to our gene, which does not give us much confidence in it. We tried to search for the protein family in literature about our gene and the transmembrane protein from Arabidopsis thaliana, but we were unable to find any more information about the gene's function.
We then studied a high-impact nonsense mutation in our gene that was found at position 1116 in the coding sequence in exon 2. And this was a mutation from guanine to adenine, and it's shown in red on this segment of our gene map.
In the normal gel electrophoresis in the left, there is a bright band at the bottom which shows the RNA contamination. With the next gel that was performed, it was performed with RNase-infused agarose, which allowed for RNA to be degraded. There were some problems with the ladder that were experienced throughout the lab, which can be seen by the shorter ladder. To counteract this, we could have ran the gel for a little bit longer.
We then used the nanodrop to determine the DNA's purity and also the concentration of the DNA that we extracted. Our 260 over 280 absorbance value showed that there was some protein contamination, given that the ideal range would be between 1.5 and 2, and all of our values were slightly above this. And our absorbance for the 260 over 230 was normal and expected for some of them. And then for the wild type and mutant 5, we had slightly unexpected values outside the ideal range, indicating that there could be some sort of salt or chemical contamination. And our DNA concentration was sufficient for us to continue with the experimental procedure.
As expected, our gene is heterozygous, so the first 2 images are expected and within the relatively minimal noise. However, we found that we were unable to conclude the sequence in the last picture because it does have a fair amount of noise and is quite unclear.
We then used staining to try to see how the mutation affected the phylogeny of the plant, specifically in its stem. We were unable to get quality images because our slices of the stem were too thick. So in the future, we could avoid this by using a machine to cut thinner slices. That way we could get better images. Based off these images, it's not clear if there is a significant difference in the stem of the mutant versus the wild type plant, so we can't conclusively determine the effect of the gene on this aspect of the plant's phylogeny. Phenotype.
Using the EFP browsers, we have confirmed that gene expression is higher in the stems and roots in comparison to the leaves. Additionally, we learned from the photocycle data that gene expression was found to be higher during the day in comparison to the night. This implies that the light variation may have an impact on our mutation or pertains to its function.
We then conducted some t-tests to determine the difference in gene expression for different treatments. So we found that there was no significant difference in gene expression for an ammonium-treated plant versus a nitrate-treated plant with a t-value of 0.0243. And we also found that there was no significant difference in gene expression based off the photocycle data with a t-value of 0.373. However, when we studied the gene expression in the leaves versus in the roots, we did find a statistically significant difference of 0.033, which is below the cutoff value of 0.05. And as you can see from the graph on the right, the root had much higher gene expression overall, and it also had more variation in gene expression, whereas the leaves had much lower gene expression.
For our experiment, we started with a total of 6 plants, with 3 set for the control and 3 set for experimental data. The experimental treatment was 10 micromolars of IAA, which is a growth hormone. However, to balance the ethanol present in the IAA solution, we made a 10% solution of ethanol for the control group. For each plant, we sprayed 8 puffs of their respective solution and let it sit for 3 hours. After the 3 hours were over, we collected the leaves and stems for RNA extraction.
So we obtained significant concentrations of RNA to continue with the procedure and conduct qPCR. And we ran a gel to determine if the concentrations of RNA were consistent. As you can see, there was, based off the dye brightness, there was more RNA in the first 2 lanes and in this 5th lane than in these middle lanes, which could just be experimental error since we had some trouble with the dye sticking to the sides of the Eppendorf tube. And so it's very possible that we didn't fully transfer the RNA in the dye into each well.
From our qPCR data, we know that a high CT value traditionally means low gene expression. However, since the CT value alone does not tell us sufficient information about the expression, we ran some statistical analysis on the data, which is presented in the slide. The mutant had higher gene expression than the wild type, and the control had a higher gene expression than the treatment group. Although our results are strange, the gene appears to be involved in some way to plant growth. IAA had decreased expression of our gene of interest, and it is possible that the gene regulates or limits growth in some way, but we still cannot definitively state the gene's function. The graph shown is in logarithmic scale because it otherwise does not allow us to see the other bars due to such a large difference.
So to summarize, our protein is most closely related to a protein from Oryza sativa. We were unable to determine the function of our gene despite looking into our gene and trying to find literature about it and also researching a transmembrane protein. So all we were able to say is that it's related to a transmembrane protein from Arabidopsis, and so it may be a transmembrane protein, but we don't have sufficient evidence to state that conclusively. Our qPCR found that our mutant had higher expression of our gene of interest than the wild type, and that IA-treated plants had lower expression of our gene of interest, which is interesting because IAA is a plant growth promoting hormone. So our gene is most likely involved in plant growth in some way based off our experimental results, perhaps regulating it because IAA treated plants had lower gene expression. And this would also be consistent with our finding that the highest level O expression occurred in the roots, which may make sense because it may be a transmembrane protein.
Lastly, we would like to thank Dr. Verma and the TA Ahmed Ali for their tremendous support in helping assure our lab run smoothly. Additionally, we would like to thank the Hazen Lab for providing us with the plans and for conducting our qPCR.